Hello everyone, welcome to the channel Microbial World. So today we will be discussing about fluorescence microscope. So the fluorescence microscope, it is a light microscope that works by the principle of fluorescence. What is fluorescence? Now here we will be staining the specimen. Okay, we will stain the specimen with fluorescent dyes called fluorochromes. Now, some of the commonly used fluorescent dyes are acridine orange, auramine, rhodamine. So, when these fluorochromes, when they absorb light, the fluorochromes will get excited and the molecules will move to a high energy state. So, here you can see this is the electron in an atom. Now, the electron, it is in ground state, which is the most stable state. When the electron, when it absorbs light, okay, the electron will get excited and the electron, it will move to a high energy state. But this high energy state, it is very unstable. Okay. So the electron, it will drop to a stable low energy state and then the electron will return back to the ground state. Okay. So when the electron return back to the ground state, it will release energy which is in the form of light. This is called fluorescence. Now, we can say that a substance is fluorescent when it absorbs energy of shorter wavelength radiation such as UV light and emit longer wavelength radiation of visible light such as green or red light. So, we can say that substance is fluorescent. Now, this phenomenon, fluorescence, it is used to detect microorganisms, antibodies and many other substances rapidly. And if the fluorescence microscope, it is used to detect antigen-antibody reaction, then it is called immunofluorescence. Okay. Components of fluorescent microscope. So, the components, it include light source. Xenon arc lamp and mercury vapor lamp, it is a common source of UV light. Okay. And in advanced forms, there is power LED and lasers are also used. Then there is excitation filter. So this excitation filter, it will only allow specific wavelength of light to pass through. So these filters, it will select the wavelength to excite a particular dye within the specimen. And it will only allow that wavelength of light to pass through. Then there is dichroic mirror, or it is also called dichroic beam splitter. This is a color filter and it will selectively pass small range of colors while reflecting other colors. Then emission filter. So the emission filter, it is a filter that will pass only wavelength emitted by fluorophore and block excitation light. So we have said that we will be staining some of the specimens with fluorescent dyes, right? Fluorescent dyes called fluorophore or fluorochrome. So these fluorophore, it will absorb shorter wavelength radiation and they will emit longer wavelength radiation or long wavelength light rays. So this emission filter, it will only allow that long wavelength light rays to pass through and it will block any other residual excitation light. So these are the main components of fluorescent microscope. You will understand its functioning more when we explain it in the working mechanism. So now let's see the working mechanism of a fluorescence microscope. Okay. So in order to observe a sample through fluorescence microscope, that sample, it will be labeled with fluorescent dyes. We have said that earlier, right? We will label that sample with fluorescent dyes, okay, known as fluorophores or fluorochrome. Okay. So here there is the light source. So the light source, it will generate a high energy short wavelength light, okay, light source such as mercury vapor arc lamp. And that light, it will pass through the excitation filter. So we said that the excitation filter, it will only allow specific wavelength of light to pass through it, okay. So here the excitation filter, it will only allow short wavelength of light to pass and it will remove all the other non-specific wavelength of light, okay. And the light will pass through and the dichroic mirror, this is the dichroic mirror, it will reflect that light. It will reflect the light and the objective lens, it will focus that light onto the fluorophore labeled sample. And then the fluorophore, we said the fluorophore, it will absorb, okay, it will absorb the short wavelength light rays and it will emit longer wavelength light rays, okay. So this is the long wavelength light rays, and then it will pass through the objective lens, and then the dichroic mirror will transmit that light and it will reach the emission filter. So the emission filter, we have told earlier, the emission filter, it will only allow the light that is emitted by the fluorophore to pass through it, okay. So here they absorbed short wavelength light rays and the fluorophore emitted long wavelength light rays, right. So these long wavelength emission will only pass through the emission filter and it will block all the other residual excitation light. Then it will pass through the ocular lens into the detector. And the image formed, okay, how the image will be? The image will be glowing images against dark background. Okay, so this is the working principle of fluorescence microscope. The advantages of fluorescence microscope is that it is more efficient, okay, it is more efficient when compared to other optical microscopes, and it is used for in vitro and in vivo imaging. It is also used to study the dynamic behavior exhibited by live cells, and they also produce images with high resolution and clarity. The disadvantage is that phototoxic effect. Okay, so we said that we are using these microscopes to analyze live cells, right? So we said we will be labeling the samples with fluorescent molecules, that is fluorophores. So these fluorescent molecules, they will generate reactive chemical species when exposed to light. Okay, they will generate reactive chemical species when they are exposed to light, and this will result in phototoxic effect, and this can affect the cells. And this microscope, it will only allow the observation of specific structures labeled for fluorescence. Then photobleaching. Photobleaching means these fluorophores, when it is exposed to light for a long time, they will lose their ability to fluoresce. That is called photobleaching. This is another disadvantage of fluorescence microscope. Applications of fluorescence microscope. Acid-fast bacilli in sputum or cerebral spinal fluid can be detected, okay, when it is stained with fluorescent dye such as oramine. And also it also helps to detect Trichomonas vaginalis. This is an intracellular gonococci. It helps to detect Trichomonas vaginalis when it is stained with the fluorescent dye acridine orange. And it also helps in immunodiagnosis of infectious diseases using direct and indirect antibody techniques. And immunofluorescence, it is useful in detecting diseases such as syphilis and rabies. So these are the main applications. So this is all about fluorescence microscope. Thank you.